Hello, I'm Owen Puls, a bioimage analyst at HHMI Genelia Research Campus, and I'm excited to highlight our recent manuscript, where we redefined colocalization using phasor analysis. Colocalization studies measure where and to what extent biological structures share similar space. In a two-color image, we want to know the following. How many pixels contain a mixture of blue and pink? In what proportion are they mixed? And where does the mixing occur in the image? Traditionally, two different metrics are used to try and quantify different aspects of colocalization, Pearson's and Manders. In reality, they merely infer it by proxy. As we showed, this means they are prone to providing unintuitive, misleading, or indeterminate results. What's worse, they provide no spatial information regarding where this mixing occurs or varies. To fill these holes, we needed more information than simply intensity. Multispectral imaging offers just that by collecting lambda stacks that contain spectra at each pixel. Phasor analysis then translates each spectrum into a pair of numbers, G and S, which can be plotted as a phasor point. If a lambda stack contains just one color, it maps to a phasor plot like this, or maybe like this. If instead, though, it contains a mixture of two colors, it maps to the mixing band. This plot contains some points which are only blue, some that are only pink, and some that are a mixture of the two colors. Phasors exhibit two important properties. First, they allow us to calculate how much blue and how much pink make up any phasor point in the mixing band, simply by asking where along the line connecting them it is. This is the basis of the phasor mixing coefficient, PMC. PMC is made up of two values, mu, which measures the average extent of mixing, and sigma, which measures the homogeneity of that mixing. We demonstrated that these two values capture biological subtlety that Pearson's and Manders often obfuscate. However, we went even further. The second important property of phasors is that they exhibit reciprocity, meaning that each phasor point corresponds to exactly one pixel. We defined a second output using this, mapping the mixing back onto the image to create the color mixing image. This is a powerful tool, which visualizes precisely where mixing occurs in the sample, a tool that other colocalization metrics simply don't provide. We validated PMC using examples of expected association, quasi-complete, negligible, and moderate. This last example highlighted the diversity of mixing that the CMI can visualize. Importantly, we showed that PMC is less sensitive to experimental choices and factors such as intensity threshold application and noise. Moreover, we also illustrated that PMC can facilitate mixing analysis even in the presence of significant background signal. These all represent factors against which Pearson's and Manders are particularly sensitive. In this way, PMC offers users an innovative, robust, and flexible way to measure and visualize biological association. I hope you check out our manuscript for more details, and thank you so much for watching.